All, this is Dr. Mubin Sayed from DrBeen.com. Welcome to one more show. So the discussion today is that can the beta cells be reactivated? Can type 2 diabetes mellitus be reversed? So the traditional medical approach to type 2 diabetes mellitus, especially in the early phases, even in the late phases, is chronic progressive disease. It is continuous and it is going to keep deteriorating. So the management approach is to continue to give drugs that would reduce insulin resistance and try to protect the beta cell from dying by reducing stress on them, let's say carbohydrates. And then finally, as they continue to die, then the person becomes insulin dependent, and then try to control further the damage done by the hyperglycemia and the glucose or glycation that happens of the tissues. However, if you look at the latest studies, for a long time now, there are studies that are showing that beta cells of the pancreas, the cells that are making insulin, are not necessarily dead. They are not all necessarily dead. So there is a deficit in the pancreatic function. There is a reduction in the pancreatic function. That means there is a reduction in the functional beta cells, and it is thought that they are dead. They have caused apoptosis or they have killed themselves. The latest research shows that a majority of them have become de-differentiated. They have stopped working due to the stress on them and can re-differentiate into active beta cells if the stress is reduced. And a fewer number of cells have actually died or have done apoptosis. So I want to show you some studies, and the message here, the summary of the message will be that we have to reduce the stress on our beta cells, and the way to reduce stress is the following. Reduce inflammation and inflammatory triggers. Reduce carbohydrates. Carbohydrates make reactive oxygen species. Reactive oxygen species cause misfolded protein aggregates. They cause damage to other lipids. They can cause beta peroxidation of the lipids. Eventually, they cause harm to the cells, including beta cells, and that is when the beta cells under stress can start de-differentiating. Reduce inflammation. Inflammatory triggers will eventually cause immune cells to enter pancreatic cells as well and cause beta cell damage, and that damage can cause the beta cells to de-differentiate. Some of them will die. Some of them will de-differentiate. The deep fried foods. I have nothing against the fried foods. The problem is that high temperature with fats can actually cause the foods to have misfolded protein aggregates in them. Plus, if there is any carbohydrates present in the fried food, then the advanced glycation end products will be formed or age will be formed. Those products, as I've discussed before as well, will, number one, cause inflammation of the GIT, and number two, when they are absorbed into the bloodstream, they would then circulate in the body, including going to the beta cells of the pancreas and cause inflammation there and cause their functional stress, which would cause them to de-differentiate. And there are many other such factors. So let's look at some of the studies. No drawings today, just some studies to understand that can type 2 diabetes be reversed by reducing stress on the beta cells. So this is DrBeen.com. For more lectures and CMEs, you can get access to it. Now here, this is the Roux-en-Y gastric bypass provides a lifelong solution for diabetes mellitus. So this is an interesting situation that bariatric surgery, especially the Roux-en-Y and pancreaticobiliary bypass surgery, this is a kind of a surgery where a part of the small intestine is also bypassed. In this surgery, a lot of patients within a few weeks become cured from insulin-dependent type 2 diabetes mellitus. Now, with the traditional approach to diabetes, where we say that the free fatty acids that are produced because of excessive fats or because of excessive carbohydrates giving rise to fats, these fats would then go and interfere with the cellular functions, causing insulin resistance, and as a result, causing diabetes mellitus type 2. And an obese person who is going through the Roux-en-Y has life-threatening fat storage. How is it possible that in such a person, when the Roux-en-Y or the gastric bypass plus the small intestinal bypass is performed, how does this person, without yet losing a lot of weight, starts recovering from diabetes? If it is just a fatty storage, then they should not have a reversal that soon, unless all the fats are gone and the fat-related issues are gone. So here, can Roux-en-Y gastric bypass provide a lifelong solution for diabetes mellitus? So in this one, if you see here, conclusion, Roux-en-Y gastric bypass is promising option for lifelong treatment of type 2 diabetes. And if I go down, there is a very interesting study they have presented. Here, if you see, Roux-en-Y gastric bypass is frequently reported to achieve glycemic control and even cure type 2 diabetes in obese patients by reducing insulin resistance. Case reports have also documented remission of type 2 diabetes in non-morbidly obese individuals undergoing bilio-pancreatic diversion for other indications. As such, a proximal intestinal bypass, that is early intestine, could be considered for the treatment of diabetes, and potentially undiscovered factors from the proximal bowel, the early intestine, may contribute to the pathophysiology of type 2 diabetes. Then look at this. Alexandres and colleagues reported resolution of type 2 diabetes in 89% and 99% of the patients following Roux-en-Y gastric bypass and BPD, bilio-pancreatic diversion, respectively. 89% and 99%. And they haven't yet lost their weight, but they have started recovering. Similarly, if you see here, this is gastric bypass surgery leads to long-term diabetes remission. And this one is printed in Endocrine Society. And here again, 57% of participants with diabetes achieved remission after gastric bypass surgery, and 22% reached remission after lap band surgery. Then if I go in here, diabetes remission status during 7-year follow-up of the longitudinal assessment, this is 2021. And here they are saying diabetes remission occurred in 57% after Roux-en-Y and 22.5% after LAGB. So again, gastric bypass. The important thing in here is that before significant weight loss is achieved, the diabetes reversal is achieved. So if fat was the one, or the fatty tissue was the one to be blamed, then this would not happen. That doesn't mean that the fatty tissue producing inflammatory hormones does not cause inflammation. It does. And that doesn't mean that the free fatty acids do not have a role. They have a role as well. But there is more than just this. And you would see in the next studies, it is really the redifferentiation of the pancreatic cells, beta cells, as they become less stressed. So let's see this. Beta cell deficit and increased beta cell apoptosis in humans with type 2 diabetes. So I have picked up this study. This is 2003. So earlier studies, even last century studies, they showed that pancreatic deficit would occur. That is, the tissue that is functional in the pancreas in diabetic type 2 diabetic patients, that functional tissue will reduce. So the conclusion was that these cells have died and they have caused apoptosis. Apoptosis means cell death, willingly dying. So here if you see, they have shown various studies and various people and their tissue. So obese humans with IFG and type 2, that is the fasting glucose, and type 2 diabetes had 40% and 60% deficit. So their pancreatic tissue. And lean cases of type 2 diabetes had a 41% deficit. So this is where we started, or these kind of studies, they started us on the path of saying beta cells commit apoptosis, they die, and that is how the deficit occurs, and that is how we end up with a chronic progressive disease. These cells continue to die. As they continue to die and the remaining cells are less, and as we keep asking them to work harder, they would keep burning out as well, and they would keep dying. So then, this one, 2013, beta cell dedifferentiation in diabetes is important, but what is it? So here they're talking about glucotoxicity, lipotoxicity. If you look at Dr. Barbara E. Corky's Banting Award lecture in 2011, she talked about redox imbalance, reactive oxygen species by the food preservatives, food colors, food emulsifiers, 4,000 food agents, especially in the ultra-processed foods, ultra-heated foods that produce a lot of reactive oxygen species that put stress on the beta cells, causing them to dedifferentiate, causing them not to function more than dying. So dedifferentiation, here they have a diagram where a beta cell, this is a mature beta cell, under the stress by inflammation or carbohydrate reactive oxygen species or other reasons for oxidations, they cause the beta cell to go back towards their multipotent progenitor, that is their parent state. Now they've gone, they have quit their function. They're not dead. They quit their function and they said, sorry, I'm not going to work anymore. I'm going to assume the role of my parent and I'm going to say, I don't make insulin. Don't put stress on me. However, if you remove the stress on them, then they can redifferentiate and start functioning again. So this is that study. So here if you see, studies looking at maximal stimulation in subjects with type 2 diabetes and controls using glucose infusion followed by stimulation with arginine or isoproterenol found that insulin response in type 2 diabetes was only about 15% of that of control, healthy individuals. Because the average reduction in beta cell mass in type 2 diabetes is about 50% of normal, it can be estimated that secretory output of a given mass of beta cell in the presence of hyperglycemia, the stressful state, might be reduced to about 30% of normal. There is a good reason to think that this loss of function is reversible because of the remarkable restoration of GSIS seen after bariatric surgery for type 2 diabetes mellitus and the improvement of insulin secretion seen after intensive insulin treatment in number of studies. Then let's look at this one. This is 2016. We are coming forward and more studies are talking about dedifferentiation and redifferentiation. Evidence of beta cell dedifferentiation in human type 2 diabetes. And here, by these criteria, dedifferentiated cells accounted for 31.9% of beta cells in type 2 diabetes mellitus. The data supports the view that pancreatic beta cells become dedifferentiated and convert to alpha and delta-like cells in human type 2 diabetes. These findings should prompt a reassessment of goals in the prevention and treatment of beta cell dysfunction. And then here, a brief review of the mechanism. So look at this. This is 2021. We have come now in more contemporary time. A brief review of the mechanism of beta cell dedifferentiation in type 2 diabetes. And here they talk about how the dedifferentiation of the cells occur. Look at this one. Similarly, a recent study showed that beta cells in the type 2 diabetes mellitus patients lose their identity and cell mass, possibly even gaining features of other islet cell types, indicating that islet remodeling with dedifferentiation is underlying cause of beta cell dysfunction during the development of type 2 diabetes mellitus in humans. Despite significant alteration in beta cell differentiation phenotype, that is the type of their work, and decrease in beta cell mass in humans type 2 diabetes mellitus, the apoptotic rate of beta cell remains relatively low. So this is a very important statement. It actually flies in the face of saying beta cells are dying. They're saying that the number of cells dying are lesser and the number of cell dedifferentiating or becoming dysfunctional are more. Why is this important? Those cells that are dead, we cannot bring them back. But those who have become dysfunctional, we can reactivate them. That is how type 2 diabetes mellitus can be reversed. That is what we are seeing with the bariatric surgeries, for example, or intermittent fasting, for example. Collectively, growing evidence suggests that beta cell dedifferentiation may be a potential mechanism for the loss of beta cell function and mass during the development of type 2 diabetes mellitus. Of course, if the cell has dedifferentiated into a different kind of a cell, the progenitor cell, the parent cell, and if you try to measure the beta cells in that tissue, you're going to say that there are less beta cell. And where did they go? They died. You can't measure the parent cell to say this is a beta cell because their properties become different. The previous study that I showed you, they actually proved that the beta cell can dedifferentiate into their progenitor cell, the parent cell, change their properties as well, so they're not bothered anymore. And then as the stress goes away, they can redifferentiate back into the beta cell and be counted again. Collectively, growing evidence suggests that beta cell dedifferentiation may be a potential mechanism for the loss of beta cell function and mass during the development of type 2 diabetes mellitus. What kind of mechanistic triggers, factors? Glucotoxicity, lipotoxicity, lipids, inflammations can do it. And here they go into a detail of how dedifferentiation can occur. So, for example, inflammation of various kinds, oxidative stress by either increase oxidative agents, carbohydrates, food colors, emulsifiers, preservatives, or by a reduced tendency or capacity of our cells to fight with the reactive oxygen species. Interestingly, beta cells have a lesser capability of fighting with the reactive oxygen species. Their antioxidants are less. So they are especially vulnerable to reactive oxygen species. So oxidative stress is a consequence of an imbalance between cellular antioxidants, defenses, and the production of reactive oxygen species such as hydrogen peroxide and superoxide. It is well known that oxidative stress reacts with the cellular molecules such as proteins, lipids, and DNA, causing misfolds and damage. Pancreatic beta cells are particularly susceptible to ROS due to their relatively low expression of antioxidant enzymes. Then ER stress, endoplasmic reticulum stress. The endoplasmic reticulum are compartments in the cell that are helping make various proteins. And when there is a lot of stress on a beta cell to make a lot of insulin, or there is a lot of oxidant stress, the beta cell responds to the irritation by producing insulin. 50% of the proteins formed in the endoplasmic reticulum of a beta cell are insulin molecules. When you irritate these cells or put stress on these cells through the mechanism of reactive oxygen species, inflammation, carbohydrates, etc., these cells tend to make more insulin, and when they are filling their endoplasmic reticulum with a lot of proteins processing in them, that stress can cause signals to the DNA of the beta cell to say, you know what, too much stress, let's get back and become dedifferentiated so that we don't have to take this burden of so much work. So here, accumulation of unfolded or misfolded proteins in the endoplasmic reticulum space overwhelms the ER capacity, resulting in the ER stress. During ER stress, downstream signaling pathways known as the unfolded protein response are activated. We have talked about it. Heat shock proteins are one, then the ubiquitination systems are another, then the autophagosomes or autophagy systems are another. So there are various kind of healing pathways that can become activated, and if they fail, the stress would start. UPR restores ER homeostasis by attenuating protein translation and alleviating the accumulation of misfolded proteins. So they stop producing, they give signals to DNA to say no more proteins yet, we are already filled here. And then they try to repair the ones, the garbage and trashy, not trashy, the trashed misfolded proteins that are there. Promoting ER-associated protein degradation to remove misfolded proteins and activate signaling pathways that increase chaperone production, thereby attenuating ER stress. So heat shock proteins are called chaperones. These chaperones are going to try to fix these proteins or ubiquitinate them to take them out and get them killed or removed or destroyed. Within beta cell, insulin production and secretion depends on the capacity of the endoplasmic reticulum. It is well known that the synthesis of insulin in pancreatic beta cell accounts for up to 50% of the total protein synthesis. Growing evidence suggests that the failure of the adaptive UPR, unfolded protein response, in the ER of beta cells is linked with beta cell dedifferentiation and loss of beta cell function. And then they continue on with more as well. The point of all of this: reduced carbohydrates, reduce the stress on the beta cell. Whichever way you are adding stress to the beta cells, that may be your own day-to-day stress, that may be pollutants, that may be impure foods, that may be the type of foods. So I am not against any one kind of food in favor of another kind. It is really whatever is causing stress on the beta cell would need to be reduced. Carbohydrates are a general culprit. Reactive oxygen species producing agents is another. Beta peroxidation is another. Advanced glycation and products or aged products are another. Deep fried foods or high temperature cooking is another. Industrially processed foods with the preservatives, colors, and other agents in them is another. Emulsifiers that try to mix, that are used to mix the fatty and watery substances like in ice creams together, these are another. So people think that I'm against French fries, for example, because I did a video about that. That's not the case. The foods that cause oxidations or produce a lot of reactive oxygen species, whatever those foods are, or foods or lifestyles that cause inflammation, will do this. So one, we should reduce them. Secondly, we should improve our defenses. Antioxidants of water-soluble type like vitamin C, of lipid-soluble type, for example, vitamin E, morning walk, near infrared light, being near plants so that the phantom sites from the plant, the essential oils of the plants would help, mitochondrial health. So all of those things would contribute, but we can get our beta cells to start functioning better again. And if they have given up and de-differentiated, we can help them redifferentiate and start working again as well. So with this, thank you very much. And I would see you again. Please like, subscribe, and share. And there are links in the description if you would like to support this work. Thank you. Stay healthy and stay free of diabetes. I'll talk to you later.